Metabolism encompasses all the processes and pathways needed for cell survival. These processes can be classified as catabolic processes or anabolic processes. Catabolic processes involve the breakdown of dietary components with the purpose of producing energy, most likely NADH and FADH2, which are eventually converted to ATP, a more readily usable form of energy. Anabolic processes will use the ATP produced from catabolism to synthesize cellular components like proteins, carbohydrates, lipids, membranes, nucleotides, nucleic acids, and other macromolecules, as well as maintain other cellular processes. Metabolism is compartmentalized. Some of the most important anabolic pathways occur in the cytoplasm. This includes glycogen, fatty acid, triacylglycerol, cholesterol, and nucleotide biosynthesis, as well as amino acid biosynthesis. On the other hand, some of the most important catabolic processes occur in the mitochondria, including pyruvate dehydrogenase, TCA cycle, fatty acid beta oxidation, and glutamate dehydrogenase, with glycolysis, glycogenolysis, and HMP pathway occurring in the cytoplasm. It is noteworthy that the urea cycle and gluconeogenesis start in the mitochondria and continue in the cytoplasm. There are three stages of catabolism. Stage 1, which occurs primarily in the intestinal lumen, involves the digestion of dietary components, polysaccharides, proteins, and triacylglycerols into their building block components, glucose, amino acids, fatty acids, and monoacylglycerol, respectively. Glucose and amino acids are picked up by the enterocytes and eventually released into the bloodstream, whereas digested lipids, fatty acids, and monoacylglycerols are reassembled into a chylomicron triacylglycerol-rich molecule and then released into the bloodstream. After their pickup from circulation, glucose, amino acids, and fatty acids are catabolized inside the cell, either in the cytoplasm or the mitochondria, to produce high-energy molecules. Of the four major catabolic pathways, only glycolysis occurs in the cytoplasm, whereas pyruvate dehydrogenase, the TCA cycle, and fatty acid beta oxidation occur inside the mitochondria. The NADH and FADH2 produced by the three mitochondrial catabolic pathways will be present in the mitochondrial matrix and will be directly accessible for the third stage of catabolism, which occurs inside the inner mitochondrial membrane. The purpose of the third stage of catabolism is to convert the high-energy molecules NADH and FADH2 into a usable form of energy that can be readily used by the cell, namely ATP. This occurs through the coupled process of electron transport and oxidative phosphorylation. Glucose has at least 4 uses in the cell. The first one is through glycolysis to produce energy and 2 molecules of pyruvate. The second one is in storage of glucose as glycogen, which occurs in both liver and muscle. The third one is in the hexose monophosphate pathway or pentose phosphate pathway, which produces NADPH and pentose phosphate or ribose phosphate needed for nucleotide biosynthesis. The fourth important use is in glycosaminoglycans, which are important extracellular matrix components, and the glycolipids, which are plasma membrane components. Fatty acids play at least 4 important roles in energy production, in energy storage, in membrane synthesis, and in inflammation. Fatty acid beta oxidation in the mitochondria is the major energy producing pathway from dietary and stored lipids. Fatty acids are stored in the form of triacylglycerol in adipocytes and provide many tissues with the energy they need during fasting. Phospholipids, the major membrane components, are made from fatty acids and glycerophosphate. The essential fatty acid, linoleic acid, is the precursor of arachidonic acid, which in turn is the precursor of all eicosanoids. There are 4 distinct uses of amino acids from dietary proteins. First one is to provide amino acids to rebuild tissue proteins that have turned over earlier. The second important purpose is to provide the carbon skeletons from amino acids to make glucose in the liver to restore blood glucose. The third important use of amino acids is in the synthesis of purines and pyrimidines needed for nucleotide synthesis. The fourth important use of amino acids is in specialized products like heme, catecholamines, and porphyrins. Cholesterol is a unique sterol and is essential for at least 4 distinct cellular functions. Cholesterol is needed for the synthesis of bile acids and bile salts, which are needed for emulsification of dietary lipids, digestion, and uptake. Cholesterol is needed for membrane synthesis and for the maintenance of membrane fluidity. Cholesterol is the precursor for the synthesis of vitamin D. And cholesterol is the precursor for the synthesis of all steroid hormones, including cortisol, androgens, and estrogens.
But this is your kind of a more traditional average diet. So 50% from carbs, 30% from fat, and about 20% from in protein. So carbs, really, polysaccharides, disaccharides, monosaccharides, they will eventually all be metabolized to give you glucose, fructose, or galactose, and those can be metabolized further. The majority, the 90% of dietary fat is tags. Okay? You gotta know that. Not cholesterol. Tags, triacylglycerol. That's 90%. The other 10% are these here: fatty acids, phospholipids, cholesterol. I'm just gonna try to connect the dots for you here for the major metabolic pathways. If you have glucose coming from carbohydrates, you're gonna have glycolysis, pyruvate dehydrogenase, TCA. That's the catabolism of glucose. All energy producing pathways. Fat in the diet, which is, as I said, tags. You break down the tags, you get fatty acids, and you get fatty acid beta oxidation. And fatty acid beta oxidation produces energy as well as acetyl-CoA, and acetyl-CoA can get into the TCA cycle and give you more energy. So I just want you to kind of connect the dots here and appreciate some of the things we've covered.
Glucose, a 6-carbon monosaccharide, is catabolized sequentially by glycolysis, pyruvate dehydrogenase, and the TCA cycle to yield 8 ATPs, 6 ATPs, and 24 ATPs respectively, for a total output of 38 ATPs. Dividing 38 ATPs by 6 carbons in glucose will give you 6.33 ATPs produced per CO2 produced. Beta oxidation of fatty acids is the major catabolic pathway that produces energy from dietary triacylglycerol or stored triacylglycerol. Starting with palmitate, a 16-carbon fatty acid, beta oxidation will sequentially remove 2 carbons at a time in 7 cycles to produce a total of 8 acetyl-CoAs at the end. The total net ATP output from the complete oxidation of palmitate to CO2 is 129 ATPs produced, which is equivalent to 8.06 ATPs produced per CO2 produced. The complete oxidation of glucose through glycolysis, pyruvate dehydrogenase, and TCA cycle produces a maximum of 38 ATPs. Dividing 38 ATPs by 6 CO2s produced gives you 6.33 ATPs produced. Complete oxidation of palmitate, a 16-carbon fatty acid, through beta oxidation gives you 129 ATPs. Dividing 129 ATPs by 16 gives you 8.06 ATPs produced per CO2 produced. The complete oxidation of acetoacetate through the TCA cycle produces 24 ATPs. Divided by 4 carbons produced gives you 6 ATPs per CO2 produced.
So we're gonna go into the specific effects of insulin, and first on carbohydrates. So there are really a variety of things that get activated, a variety of things that get inhibited. What gets activated is glycogen synthesis and glycolysis, two processes that consume glucose because there's plentiful glucose in the bloodstream. Okay? The reverse processes, glycogenolysis, which produces glucose, and gluconeogenesis, which also produces glucose from smaller molecules, are inhibited. And that's specifically important in the liver, because the liver is the only tissue that has gluconeogenesis. In both liver and muscle, you have a decrease in glycogenolysis, degradation of glycogen. The tissue-specific effect of insulin is this last one. This should be really highlighted, very important. Insulin stimulates glucose uptake in these specific tissues, muscle and adipose tissue, by increasing the number of GLUT4 transporters on the cell surface, in the cell membrane. So more transporters, more glucose taken up into the cell. The effect of insulin on lipid metabolism. Let's start with adipose tissue. You remember, adipose tissue is where we store a lot of tags. The adipocyte is roughly 80% tags. Okay? So adipose tissue, important effects on adipose tissue is first to decrease lipolysis by inhibiting hormone-sensitive lipase. Second is you increase tag synthesis. And how are tags synthesized? That you need fatty acids and you need glycerophosphate. The fatty acids are coming from the dietary tags, from chylomicrons. Lipoprotein lipase breaks them down, picks those fatty acids up into adipose tissue. Then they are esterified to glycerophosphate. Where is the glycerophosphate coming from? It's coming from DHAP from glycolysis. So remember, adipose tissue picks up glucose. It's using glucose for its own energy needs. So it will have an ample supply of DHAP and glycerophosphate as a result. And that's the backbone used to esterify fatty acids from the diet or from de novo synthesis from VLDL, and you increase tag synthesis. Okay, so glucagon is going to have, again, its own set of effects on metabolism. The effects on carbohydrates primarily are to increase glycogen breakdown, glycogenolysis, by activating pKa phosphorylating glycogen phosphorylase kinase, phosphorylating glycogen phosphorylase, and that results in glycogenolysis. Okay? Simultaneously, it will inhibit glycogen synthesis. That's always very important. You're activating one thing, inhibiting another. You know, so you're not having both occur simultaneously. And the inhibition of glycogen synthesis is because you phosphorylate glycogen synthase by pKa, which becomes inactive under those conditions. You have an increase in gluconeogenesis. Okay? Both of these are key functions, again, of the liver. These are the key functions of the liver. So think about it. Glucagon is high when glucose in the bloodstream is low. That means you are in a fasting cycle. So the liver's function is going to be completely changed, meaning it has a new function. Its new function is going to be producing glucose, okay? How do you produce glucose? Through either glycogenolysis or gluconeogenesis. What happens in with lipids? You're going to use whatever tags that were stored before, either in adipose tissue or the liver, as a source of fatty acids. So you break down tags. Lipolysis will be increased primarily by activating hormone-sensitive lipase. Okay? But I want to kind of point something out. Adipose tissue doesn't have a glucagon receptor. So hormone-sensitive lipase is activated in adipose tissue by epinephrine through the beta-adrenergic receptor. Okay? So it's a unique and important kind of detail to pay attention to. So you have fatty acids are going to be used as a source of energy instead of glucose in the liver, in other tissues, in the muscle, in the heart, in many tissues that can use fatty acids, but not in the brain and not in red blood cells. Okay, these two organ and cells still need glucose as their major source of energy. Now, what's going to happen as well is that the liver will be increasing ketone body synthesis, ketogenesis, because it's using fatty acids as a source of energy, producing a lot of acetyl-CoA, and some of that acetyl-CoA is used in ketogenesis. So note how important the liver is. It's providing glucose, providing ketone bodies, two different forms of energy to other tissues that can use those forms. In terms of gluconeogenesis, what's going to happen, where the building blocks or the precursor molecules for glucose coming from? They're going to come from muscle amino acids. So in the liver, there will be increased uptake of amino acids. Again, muscle protein turnover. It doesn't mean that you're wasting away. It just means normal muscle protein turnover. Breakdown of muscle proteins gives you amino acids. Amino acids released in the bloodstream, picked up by the liver. So this is amino acids coming primarily from turnover of muscle proteins. Okay? And they are going to provide the carbon skeletons for gluconeogenesis in the form, we talked about one of them, alanine can be released, goes to the liver, becomes pyruvate. Pyruvate can be used to make glucose. So here's a summary of the effects of glucagon. Pretty straightforward. Without going through all of them, here's mainly effects on the liver. Activation glycogenolysis, inhibition of glycogenesis, activation of gluconeogenesis. This one here, note, no effects on muscle. Really, no effects on adipose tissue. This is an effect of epinephrine on the beta-adrenergic receptor that in turn activates adenylate cyclase in adipose tissue, producing cyclic A, activating pKa. pKa phosphorylates hormone-sensitive lipase that breaks down the tags in adipose tissue and make those fatty acids available to the rest of the body. Okay, so that's why it has a different color. This is due to epinephrine. If you like this video, please subscribe. Thank you.